I gotta turn the audio on. Okay. We're gonna go a little bit on what is a fiber type, how do we know what it is, how have we traditionally done this, and then we'll get into some of the functional characteristics. Okay, about why does it matter? Okay, why is this a thing that, as part of his thesis, Ty wants to understand, and that we're going to then use to predict what kind of an athlete or what kind of event some of you might be better at. Okay, so think of fiber types as a flavor. Muscles come in different flavors. Think back to the myosin that we talked about last week. I said everything comes back to myosin, the actin, the cross-bridge cycle, right? That's the root of all things in exercise. Well, fiber types come from the myosin ATPase enzyme on that myosin head, okay? And its ability to break ATP down so that we can let go, right, of the actin in the cross-bridge cycle and then start the whole thing over again. We know about muscle fiber types. We actually, in the life of science, haven't known it that long. Probably since like the 1960s, okay? And we had some idea about these things in animals, okay? Animal research is great, except that it's terrible, right? Because you have the little rats, you have the little mice, you do all these things to them, and then you just kill them, and you take their entire muscle and you grind it up, or you cut their nerves, and then it's a necessary thing as part of science. I'm glad people do it. I'm just not doing it, right? But we knew some things about fiber types, but there was an invention in the 1960s called a muscle biopsy. And specifically, the invention was from a Swedish guy named Bergstrom, and it's this specialized needle that looks like this. And what you do with the Bergstrom biopsy needle is you take a scalpel and you cut about a 1 centimeter hole, okay? You cut through the subcutaneous fat, through the fascia, you stick the biopsy needle down and in, and unlike a regular needle, nothing comes out of the end, okay? There's a cone, but that's just to help it go in. There's this little window a little bit farther up. And then there's a tube inside the tube that's really sharp, and it functions like a guillotine. And so you put the needle in, you attach one end to a plunger, you apply some suction, and it pulls muscle fibers into that window. And then you slam down the guillotine portion, and it cuts off those fibers. And then you pull the needle back out, and you get a piece that looks like this. It kind of looks like an old school pencil eraser, okay? And then some poor grad student, which I have done, you take these pieces, you basically have a fancy piece of chewing gum, you mount it on the chewing gum on a popsicle stick, and then you dump the popsicle stick into liquid nitrogen and you freeze it, okay? Then you take the biopsy, you take that little piece of muscle, and you take the popsicle stick and you put it in this outrageously expensive, it's basically like a deli meat slicer, okay? You put it in there, and you crank this thing around, and it cuts little tiny, like almost translucent, like micrometer thick slices off of the muscle, and you can put that under a microscope and you can look at the individual fibers that way. And you can take multiple slices that are right next to each other, so you see the same fibers in every slice. Some of them you can incubate with something that will tell you about the myosin. Some you incubate, it will tell you about, right, how many mitochondria it has. Other ones you can incubate that will tell you about other aspects of those fibers. And from doing that, and then the advent of biochemistry that began in the 60s and 70s, we know and understand better the different flavors that we have of muscle biopsies, okay, or of what your muscle fibers are going to look like. So the old way of doing this was something called histochemistry, okay? We would essentially bathe those little slices of muscle in different, like an acidic or a basic solution that had some stuff in there. And if it had a slow, quote unquote, myosin ATPase, it would turn one color. If it had a fast myosin ATPase, it would turn a different color. Okay. Now, the slow and fast does not come from the biopsy thing itself. The slow and fast comes from, we knew in animals that if you pulled out individual fibers, some of them would contract faster than others. And so they said there's some that are fast and there's some that are slow. And then later on, we figured out from the histochemistry, right, the ones that are slow have a slower myosin ATPase. The ones that are fast have a faster myosin ATPase. Okay, so that's where the fast and slow comes from. It's all about myosin ATPase. We could also then take those same fibers, know what the myosin ATPase speed is, and then learn about enzymes in metabolism. SDH stands for something called succinate dehydrogenase. Okay, anybody off the top of your head know where that enzyme lives? No one? That's okay. Anybody had biochemistry? The worst class in history, right? You have to memorize all the amino acids and all the enzymes and all the other things. Succinate dehydrogenase is like the enzyme that catalyzes, I think it's like the fourth step in the Krebs cycle. It's the third or the fourth step. It's not the rate limiting enzyme, but it's one there. So SDH levels tell us about mitochondrial function, Krebs cycle function. LDH stands for lactate dehydrogenase. This tells us about anaerobic metabolism, okay, and turning pyruvate into lactic acid. Hexokinase is the first enzyme in glycolysis. It puts a phosphate on glucose and traps glucose in your cell. And so again, we can stain the fibers for these enzymes and figure out which ones have more or less. If a person is more or less trained, do they have more or less of all of these things? And that's how we know a lot of stuff about all of this. You could also, with histochemistry, stain them for muscle glycogen. You could stain them for what we would call intramyocellular fat. Okay, so which fibers have more glycogen? Which fibers have more fat? How is that going to play a role in metabolism? What are we going to use for energy and a host of different things? So we learned a whole bunch in the 70s doing this. In the 80s and kind of the late 70s, we did an advent of something called immunohistochemistry. How many of you, probably shouldn't ask for a show of hands, how many of you have taken like an at-home COVID test? Or the new at-home COVID and flu and all of those kinds of tests, okay? You swab your nose, you think you're stabbing yourself in the brain, you pull it out, you put it on there, and it runs down the chromatography paper, right? Those things use what we call immunohistochemistry. There are antibodies that react with pieces of the COVID virus or pieces of the flu virus or whatever you want it to be, okay? And so we have specialized in antibodies now that will react to specific proteins on the muscle fibers. And the one that we tend to use is specific for something called the myosin heavy chain. I told you guys what the myosin heavy chain was last week. It's kind of one of the backbone pieces of the myosin head, but this MHC or myosin heavy chain comes in different flavors or different isoforms, and it corresponds to the myosin ATPase speed. And so we use the heavy chain now when we do any kind of fiber typing on those things. Okay. Histochemistry gave us fast and slow. Immunohistochemistry taught us that there's not just fast and slow. There is slow, and then there's multiple flavors of fast. Okay. And so this gives us slow twitch, fast twitch. Immunohistochemistry gives us type 1, type 2A, type 2B, type 2X, type 2C, embryonic myosin. Okay, it lets us be a little bit more specific about things. All of the differences between the fiber types occur on the fast twitch side. There's one kind of slow twitch fiber and only one. Only one. Okay, all the rest of the differences on the fast side. Okay, so we're going to talk about things for the most part using this naming convention. I'll talk about type 1, we'll talk about 2A, we'll talk about 2X. These are the three primary human skeletal muscle fiber types. Okay. If you're super sedentary, you might have some type 2Bs. Maybe there's some 2Bs. Okay. But for the most part, you guys don't have 2Bs. Rats, mice, cats, they have type 2Bs. That's where the naming convention came from, was from those lower animals. They're even faster. Okay, so we typically now talk about 2X. There also exist these hybrid fibers that are kind of transitions, if you will, between 2A and 2X. And there's a host of them. There's 2AX, and then there's 2XA, which is kind of like, right, you've got north, northwest, north, northwest, northwest, north, like you have the little things on your compass. They're going to denote these transitions in between. Okay. Most of you guys, most, have very few type 2X fibers. Okay. Most of your fast switch fibers, of which many of you have a bunch, most of your fast switch fibers are 2A. Okay. If you're at least moderately active, most of your 2X fibers will become 2As. And we'll explain a little bit more about the how and the why when we get into endurance training, but that's what happens here. Okay. Your percentage of type 2 versus type 1 fibers is determined at birth. It is genetic. You have no control over it. Okay? None. Almost every muscle in your body has a relatively similar percentage of type 1 to type 2, the ratio of those to each other. Most of you are about 60-40 one way or the other in most of the muscles in your body. In all of us, our diaphragm is the slowest or the most type 1 muscle in our body, right? Very, very fatigue resistant. Your gastroc in your soleus, okay, postural muscles in the legs that we use all the time, probably the next slowest or the next most type 1. But most of us are about 60-40 one way or the other, okay? And you can't change that. You cannot change that. No amount of training, no amount of wanting, nothing will change that. Unless you let me cut the axons from your motor neurons that go to those muscle fibers and swap them around, then I can do it. Okay? Then I can do it. I had a buddy, that's what his postdoc was about. They would cut the nerves in rats and reattach them and move them around, and it will change the fiber type on some of those things. Okay? Okay. Here's just a picture of, in some ways, what the histochemistry used to be. You're not responsible for this. You don't have to know, but you will just note, like we're bathing fibers in an acidic pH. We're bathing them in a basic pH, and different ones will turn different colors. And that's kind of how we know whatever it is that we're going to do. When we speak of fiber types now in today's world, we don't just talk about myosin ATPase speed. That gives us a good picture, but we also then talk about what are its functional properties. Is it fast? Is it slow? How quickly does force turn on? How quickly does force turn off? How many mitochondria does it have? What does its metabolic profile look like? Okay, because all of those things are important. Because you can have a lot of type 1 fibers, which in theory metabolically are better from an endurance standpoint, and you can be a shitty endurance athlete because you don't train. You have no mitochondria in all of your type 1 fibers. And someone else might be almost exclusively type 2, but they run or they bike a bunch. They have a lot of mitochondria, and so they're a better endurance athlete than you are. That part we can train. But the actual sort of ratio of fast to slow or 1s to 2, we can't really do a whole lot about all of those. So we want to look at this, okay? Also, we need to understand mitochondria, how many capillaries do you have going to those fibers, how much myoglobin do you have there, and we'll get into capillaries and myoglobin when we get into the cardiovascular system, but it's a more complicated system than just fast versus slow, okay? Does that make sense, guys? You all with me on all of that, okay? Now, one of the easiest ways for us to know, and I guess we could have brought the Vertec in today, the seedling's high enough. I could have all of you come down and we could weigh you and then have you do a vertical jump for us and figure out roughly who can jump the highest per their body weight. Probably rank everybody that way, and it would give us a reasonably good approximation of who has more fast twitch fibers relative to everybody else. Fast twitch, you can jump high, you can run fast. Okay. reasonably well in the midst of that. That's one of the ways that we can try to know and understand these things. Okay. All right. This shows us the maximal shortening velocity, or the myosin ATPase speed, between type 1, 2A, and 2X fibers. Okay. 2X fibers are the fastest fibers, except they almost never exist. There's a thought among people that are Olympic sprinters that you train and you train and you train, and your X's get turned into A's, and then you take a break for a few weeks before your actual competition, and some of those A's begin to drift back towards 2X, and now all of a sudden you're actually faster because you've changed the underlying fiber type. I don't think we have some evidence that doesn't work as well as we would like for it to, but in a vacuum, that is a thing. Okay. The faster the shortening velocity, the better sprinter you are, the higher you can jump in some ways. Okay. So that's kind of what's happening with those. Okay. This one on the left, okay, is showing something called specific force. Specific force is a really interesting physiological measure. This is a measure of how much force we get per the size of the muscle fiber. Okay. You all probably know someone that is bigger or smaller than you, but that is also like they're bigger than you, but they're not as strong as you. Or they're smaller than you, but they're definitely stronger than you, right? From a muscle size standpoint. And so what this shows is how much force do we get per area of muscle fiber in type 1s and 2A and 2X. And so we do know two type 2 fibers give us more force per their cross-sectional area. So a person that has more type 2 fibers per their size of their muscles likely can be stronger than somebody that is all type 1. Okay. Now, most of us are pretty close in the middle, 50-50 one way or the other. So this kind of comes out in the wash, but potentially on the very extremes, this is going to be a thing. And the sprinting and jumping really comes down to this. This is a measure of maximal power. Anybody have biomechanics? Okay, only Veer. Okay. Anybody had physics 1 that you want to admit to have? Anybody have physics 2? Worst class. It's almost as bad as biochemistry. Magnets, oh God, there's everything. Electricity, magnets are all backwards, right? But you guys remember power. The equation for power, right, is force times velocity. And so if I get more force and it goes faster, my type 2 fibers have way more power. We need to apply power to sprint and jump and do things like that. That's why if you have a lot of type 2 fibers, you're a better sprinter, a better jumper, right? Things of that nature. We hope on Saturday, right, that we have football players with more power, better sprinters, better jumpers, all of this stuff than Michigan does, right? I cannot emphasize enough how much I hate Michigan. I cannot. Really, really, okay. In the next few slides, there's going to be kind of an accounting like all of this, where it's going to say, what are the fiber types, right? Oxidative capacity stands for aerobic metabolic capacity, Krebs cycle, right? Electron transport chain, right? And that is going to be related to their resistance to fatigue. Can they generate force over and over and over and over again? So type 1s, high oxidative capacity, very resistant to fatigue. That's why your diaphragm is type 1. That's why your soleus is type 1, so you can stand up, so you can breathe. Those muscles won't get tired, right? Relative contractile speed is slow. The only little piece at the end, which will make more sense once we get into the neuromuscular side of things, is something called a motor unit, or motor unit size. A motor unit is one motor neuron that's in your spinal cord and all of the muscle fibers that are connected to that one motor neuron. Okay. Type 1 muscle fibers are connected to type 1 motor neurons. And type 1 motor neurons connect to a relatively small number of fibers. So we have small motor units that have type 1 fibers. This will become more interesting and more important later on today. Okay. But the next couple of slides, it's the same thing for 2A, the same thing for 2X, right? And those things are pretty well straightforward. Okay. Questions about fiber type? Anybody have a question about fiber type? No? Got it? Lane. No, great question. There are very few muscle fibers in type 1 motor units. Okay. As you all will see when we get a little bit farther today, that every motor neuron in your spinal cord connects to a certain number of individual muscle fibers. And type 1 muscle fibers, that's called a motor unit. They're small motor units. There's very few fibers. And that has really important functional consequences, okay, about which fibers we use more often and our ability to do things like write and have fine motor skill and everything else are related to the size of those motor units, and our ability to do fine motor skills and not get tired is related to the fact that our small motor units are made up of type 1 fibers. Really important functional consequences. Okay. All right. While I get the next set of PowerPoints up and going, turn to your neighbor, introduce yourself to your neighbor, tell your neighbor where you're from, and tell them your favorite animal. Go. Talk amongst yourselves. Doing this again. Yeah, I go back. I went back last time. I am graduating. I come back from there we go. I do. I already do. Yeah. Okay. Probably. All right. Excellent. We're meeting our neighbors. You now have a friend in class, right? You know their favorite animal. So there we go. If anyone cares, my favorite animal is rhino. Very important to Ellis. Her favorite animal is panda. And so these are things that are important to 6-year-olds. So nice and fun. All right. So up till now, we've talked about skeletal muscle anatomy. Now we're going to do a little bit of nervous system anatomy, because without the nervous system and its connections to skeletal muscle, we can't make skeletal muscle work. We can't get it to contract. We can't get it to relax, which means you can't run when you want. You can't jump. You can't do whatever it is that you want to do. Okay? We are going to talk about the brain. We're going to talk about the spinal cord. All right? We are not going to talk very much about spinal nerves, but we are going to talk about motor neurons and their location within the spinal cord. Okay? And so all of this, for the most part, right, is central nervous system, is brain and spinal cord, and then parts of the peripheral nervous system are going to be this somatic piece where there are going to be axons that come out of the spinal cord and go out and connect to our muscles. Okay? So the key thing to understand right now is your brain is connected to every muscle fiber in your body. Okay? Millions of muscle fibers in your body. So you have millions of connections from your brain to your muscles. Okay? Still blows my mind. I still cannot imagine how things actually work, right? All your brain is a series of cells that just fire electrical signals. And every bit of us, what we see, what we think, how we perceive others, everything is just because these neurons are firing electrical signals. It's crazy, right? It's crazy. It blows my mind. Okay. So let's talk about those connections. Let's talk about how the brain connects to skeletal muscle and then how it uses those connections to communicate and tell muscle contract, relax. That's basically the only, it's kind of an on and off switch. Okay. We signal from the central nervous system. You have, well, I guess the motor cortex is up here, I think. Again, no anatomy. I don't remember. Right? Right? Motor cortex, anybody? Front? I feel like it's sort of up here.
It's the front.
Yeah, front part right here. Motor cortex. Okay. Your motor cortex connects to neurons in your spinal cord. All right. And so you think about moving or something in your motor cortex, probably in your premotor cortex, here, signals down the spinal cord. In the spinal cord, there are specialized nerve cells called motor neurons. They are going to exist at very specific locations. Every motor neuron, of which there are hundreds of thousands, okay? Every motor neuron will connect to muscle fibers in a very specific muscle. And it only connects to a certain number of fibers in that given muscle, okay? So you have about 250 million muscle fibers in your body. We've got about 450,000 in an average person motor neurons. Every fiber has a connection to one and only one motor neuron. Okay? And every motor neuron, right, because the numbers, the math won't math if it doesn't go the other way. Every motor neuron connects to multiple fibers. Okay? A motor neuron and all the fibers to which it connects is called a motor unit. So what you see here, this is a motor unit. We probably don't have motor units that only connect to 5 fibers, but that's the idea, okay? One motor neuron and a pool of muscle fibers in a specific muscle. All of the fibers in a motor neuron are of the same fiber type. So if one fiber is a 2A, all the rest of them are a 2A. Okay? Here you can see then that we have what we call a motor neuron pool, or a group of motor neurons, all of which connect to the same muscle. So here we have, right, and you would have thousands of them per muscle that are going to connect, let's just say, to the biceps, okay, or the quad, or the soleus, or your glutes, or your pecs, or your tricep, or your eye muscles, or whatever, okay? So this gives us the idea of voluntary control of skeletal muscle via the central nervous system. You only contract and relax when the brain tells your muscles to do so. Even if you don't really think about, like I'm walking, you're not really thinking about walking very much, but your brain in the background is. And it is saying, signal, walk, turn this one on, turn this one off, turn this one on, turn this one off, right? We do very complicated movements. Walking requires contractions from like 40 or 50 different muscles all at the same time in exactly the right timing, okay? In exactly the right time, or you can't walk. And it all just happens seamlessly. You don't think about those things until you watch somebody that doesn't know how to do a particular task try to do it for the first time. It's great watching, we call them a baby or a toddler, try to learn how to walk or try to learn how to run, because you can really see, like in real time, like they're really thinking hard about foot here, land this way, do all of that. You guys have learned, when we just take it for granted. Boom, we just go, right? Lots of occupational therapy, physical therapy, in some ways even personal training in some of these things is we are training these connections. We are training what we call a motor path. We're training how the motor cortex signals to the motor neurons to turn on the right fibers in the right muscle in just the right order so that we can generate the movement that we want. Okay? Someone has a stroke and they get damage to the motor neurons or to the neurons in their motor cortex, and they in essence forget what those signaling pathways are. And we have to try to work with them to learn them again. Okay, to learn them again. Some of the neurons are reasonably what we would call plastic. Some can come back, some you can relearn. We have enough of them that some of them die, we can kind of take over from some others. Other times they're like, you lose motor neurons in the spinal cord, they're gone. I have fewer motor neurons than you guys do in my spinal cord because I'm older. Some of them have just died. Okay. And that's the thing that happens. It continues to happen as we age. Okay. All right. Let's do a couple of structures then on a motor neuron. Here is one motor neuron. So when I talk about a motor neuron, what we most of the time are referring to is the big portion that's called the cell body. Okay. The cell body is the thing that sits in the spinal cord. Okay. Projecting off of the spinal cord, these little finger-like structures, those are called dendrites. And they do just what you think they do. You have a whole bunch of motor neurons all by each other in the spinal cord, and all of their dendrites reach out and touch, in essence, the dendrites of the muscle or the motor neurons around them. And they talk to each other, right? Through all of that. So if some of them get a signal, they may send a signal through the dendrites to tell this motor neuron to do something. Okay? Then coming off of the cell body is this long structure here that's called an axon. When you think of nerves, okay? When you think of nerves, when you guys dissected a cadaver in anatomy and you saw a nerve, what you're really seeing is a giant collection of axons. Okay, it's a giant collection of axons. When you bump the funny bone on your elbow, what you're doing is you're bumping those axons. But axons leave the spinal cord and they're going to go all the way out. And then as they get close down to the muscle and the fibers in the muscle they're wanting to connect to, they will branch off, okay, into these little smaller structures that gives us more of them. And then they will get very close, very, very close, but not quite touch individual muscle fibers. There is no physical contact between the end of an axon and the surface of a muscle fiber. Okay? I have no idea why that's the way that it has evolved and it functions, but that's what happens. Okay? There is no direct contact. Okay? Around the axon, especially when it is sort of outside of the muscle, most axons have a fatty substance around them called myelin. Myelin is an insulator. Myelin helps us transmit action potentials faster down an axon. Okay? You think move, and it happens almost instantaneously. Well, that is because brain, think about moving, signals all the way down the spinal cord. It's electricity, so it moves fast. But then it's got to go from here. Let's just say I've got motor neurons right down here, like S2, to go to the muscles in my toes. And I step on something, I got to pull away. I want to move. It's got to go all the way down. Feet of axon, several feet. I'm not very tall, but all the way down. So we need this myelin to help us speed up. There are spots in between here where there's no myelin. Those are called nodes of Ranvier, right? High density of sodium and potassium channels at the nodes of Ranvier. And functionally, action potentials go faster because of the myelin, because they, in essence, jump from one node of Ranvier to the other. They don't actually jump, okay? It's one of the few things in physiology that does not make any sense to me, but they do sort of jump and hop down, and that speeds everything up. Okay? All of that stuff should be reviewed. You guys have seen and heard of this before, right? Okay, good. We need to understand this to understand how things are going to work. A couple of extra things about the cell body that I want you to know. The cell body has a nucleus, okay? In the nucleus, there's also a thing called the nucleolus, which has a bunch of ribosomal RNA on it. Nerves need a ton of proteins. So the nucleus has to provide all of those proteins. It uses all of the ribosomes in the nucleolus, the ribosomal RNA, to be able to do things, to make all of these proteins. Okay. Problem with the cell body is after birth, they cannot divide again. So if you get damage and something happens to them, they're done. Okay. They will die and you will lose the function from that particular motor neuron. Okay. So we have these things called Nissl bodies that are right here. Now what's called the axon hillock, which is where the axon begins to come out. Nissl bodies have lots of ribosomes, lots of endoplasmic reticulum. Basically, the nucleus says make these proteins. They get made by the ribosomes here in the Nissl bodies, packaged up in the endoplasmic reticulum. And then if you look down an axon, there's a bunch of there's a bunch of what we call cytoskeletal proteins. All right. Microtubules, things like actin and things called neurofilaments. But in essence, you make something in the spinal cord, you make proteins, like things you need, like acetylcholine and stuff, and it has to walk all the way down the axon to get to the neuromuscular junction. And so there's all of this constant stuff that is happening as we're going to move all of this. Because of this, if you damage an axon, we can repair it. If you damage the neuromuscular junction, we can repair it because of all of this capacity here to do those things. But if we get sort of lethal damage in the cell body, no mas. Okay. All right. Here we have, I don't know, somebody that's had anatomy. Is this a transverse section of the spinal cord? Anyway, I'm like this, and you just cut me right down the middle so you can see. Okay. You do not have to memorize this, but I want you to appreciate in general the location of these things. We only have motor neurons in certain locations in the spinal cord. Okay. And they tend to run from cervical down as it relates to where those muscles are in your body. Okay. So let's just say higher ones at like C3, C4, C5 are upper body motor neurons. Now, interestingly, you will note that the diaphragm is actually at like C3 and C4. That's the highest one. You're least likely to get damage to vertebrae right up here, the closer we get to the skull. So the diaphragm is up there. So you're at least not that likely to get damage to the motor neurons that go to your diaphragm. So you can at least not die from not being able to breathe. Okay? But then it works its way down. In the thoracic vertebrae, we have abdominal region muscles, like some things that are here, not a lot else. Then we get down into lumbar, and we're going to get sort of upper leg muscles, and it's going to work our way down into kind of the high levels in the sacral vertebrae. About S1, S2 is where the plantar and dorsiflexors are going to be for the things that control our lower legs and our feet. Okay? So all of the motor neurons that go to a specific muscle are in the same place in the spinal cord. They're all in the same place. So let's just say that your quad muscles are at L3. Your knee extensors are at L3. Okay? They're all at L3. So the good part of this is because they're all in the same place, you can make all of them contract or fire at roughly the same moment in time, which helps us get more force. It helps us get very good controlled movement, right? If you had some, let's just say at C6, the ones at C6 would fire before the action potential ever reaches it down here into your lumbar spine. And so you get this real kind of herky-jerky way of muscles contracting. That's the good part. They're all together. The bad part is they're all together. So if you get damage or a cut or a break or something in a particular location in the spinal cord, then you lose the function of all of those motor neurons at that one particular location. Okay? We visualize motor neurons as existing in these kinds of tubes, if you will, like a cone or a cylinder where they're all going to be stacked up together. Okay? And so you can see like my quads are here, the TA is going to be here, my glutes are here, right? There's some calf muscles. I'm moving from L4 down to S2. Yes, I know that the previous one said, right, that the quads were at L3. I don't know, it came from the same book. There's some gray area in kind of where all of this stuff is going to exist, but they're all going to kind of sit like this. Okay. This has really, really important functional consequences because they're all there together. Okay. Motor neurons, cell bodies lie in the ventral horn of the gray matter of the spinal cord. The ventral horn is the inside part, right? The dorsal horn is the back. So if you look here, okay, we have ventral horn, and then dorsal horn is going to be up there. So you've got cell bodies, axons leave, they all come out. Most of them will come out from a vertebral standpoint through the foraminal space and out, and then they all bundle up together. You will also note, though, that there are what we call afferent with an A, sensory axons leading back into the same location from those same muscles, but they come in on the dorsal side. This is really, really important because when you move, you get a sense of where is my limb in space? How much force am I generating? Am I about to fall over or not? Am I touching something hot with my leg or cold or whatever? And that way, that information can come right back into the spinal cord at the same place where the neurons that control the movement of that muscle are. And so sometimes these things will talk to each other, and it will help us adjust our position, adjust our force, and do things where it doesn't even have to go to our brain. They're called reflex. Just adjust right thing. Okay. All right. Okay. We will stop here. We will pick up on Friday. There's like 3 cookies left. Please take a cookie. All right. I will see you guys on Friday. Thank you.